Hello, good morning, good evening, good afternoon. Hola, bonjour, guten tag, ciao, namaste, konnichiwa, aloha. And for all of our participants listening from Israel, shalom. It's a pleasure to welcome you to another Global Immunotalks. I'm Susan Keck, and I'm here to introduce Professor Yardena Samuels from the Weizmann Institute, who's our speaker for today. Professor Samuels started her education in Cambridge in the UK. She then went on to do her PhD at Imperial College and worked as a postdoc fellow in Bert Vogelstein's lab at Hopkins. Her first professorship position was at the NIH, but she then moved to the Weizmann Institute in 2012 and has moved her way up to important leadership roles. She's the director of the ECARD Institute for Cancer Diagnosis and Research and is the Nell Family Professor Chair also at that institute. Professor Samuels has received a lot of awards. She's received the Pezoller Foundation Award, the Udim Family Prize in Excellence for Cancer Research, and is president-elect of the European Association for Cancer Research. And she spent a lot of her career has been focused on understanding the genetic mutations that underlie cancers, focus a lot on melanoma, and has paved the way computationally and genetically to identify what are the driver oncogenes, tumor suppressors, as well as tumor evolution and tumor heterogeneity. One of her major goals has been to delineate ideal protein targets that will achieve lasting control of melanoma through targeting some of these drivers, as well as developing and eliciting better immunogenicity towards melanoma. Her lab was one of the first to discover ERB4 as being one of the most highly mutated oncogenes in melanoma, as well as was the first to do whole exome sequencing on melanoma, which then led to, as we know, the massive and impactful repository of the Cancer Genome Atlas, of which her group contributed to a lot of the samples for cutaneous melanoma. And since then, she's been moving more and towards the immune space to try to understand what are going to be the neoantigens, where do they come from, what are going to be the best neoantigens, and using a lot of really powerful genetic and proteomic combined with computational methods. And so she's just been really paving the way for us to think about how to understand the tumor burden, tumor heterogeneity, and what that means in terms of regulating anti-tumor immunity. So it's a pleasure, Yardena, to have you here today, and we wanted to get to know you a little bit more, and so we were hoping you could share one of the most impactful decisions of your scientific career. Not that that's an easy question to answer, but could you tell us a little bit about that?
Thank you. First of all, thank you for that really beautiful introduction. I really appreciate being invited to this long list of very impressive speakers and to this initiative of Immunotalks. Regarding the question on impactful decisions during my career, right there, obviously several, but I think one of the ones that really made a huge difference is deciding where to do my postdoctoral studies, which obviously is a very difficult decision and I think a very important one. So like you said, I decided to do my postdoctoral studies in Bert Vogelstein's lab, which really paved the way to the way I chose the rest of my career. So that's where I got educated to become a cancer geneticist. That's where I developed the understanding that this is a really powerful tool to investigate cancer, and in order to identify novel targets to diagnose and to treat cancer. And so that was a very impactful decision. And so I learned lots of tools and ways of thinking, which I'm still applying basically ever since then. And I also gained a huge scientific family thanks to that experience, and an amazing mentor by Bert, as well as additional mentors within that lab. So that's probably one of the biggest impacts that I had in my career.
That's great. Wonderful. Well, we're really looking forward to your talk. So go ahead and start sharing your slides.
Okay.
All right. And we can see it. There you go. Perfect. You're in presenter mode. Yeah, there we go. Perfect.
Good. Okay, excellent.
Yep, looks great.
Okay, so I'll be talking today about the immunological landscape in melanoma. I'll give you just a brief overview on various aspects of my lab. So over the years, we started off by establishing the melanoma genetic landscape. We performed the first whole exomes in melanoma, and we're aiming to identify the drivers and passengers. We then put ourselves the goal of identifying the functional effects of these somatic mutations in melanoma. And in the last 8 to 10 years, we've been moving into the immunogenomics field, looking at the interaction of melanoma with the immune system. And in order to, and obviously all of these different names interact, and to deal with these questions, we established a very robust melanoma tumor bank, mostly in collaboration with Stephen Rosenberg at the NCI. So we have a very large number of samples and low passage cell lines that we have annotated somatically. And like Sue said, we took part in the TCGA and found that our tumor bank subcategorizes to 4 different molecular landscapes. And now we're using this tumor bank also to look at the immunogenomic landscape. And so this is just a summary slide looking at various drivers that we identified over the years, which we can now use also to understand the interaction with the immune system. So, of course, like we know, mutations will drive cancer progression. We have this evolutionary tree of the various single founder mutations and how they keep developing and affecting cancer progression. But we also remember that these particular mutations will also ultimately be involved in causing immune rejection. We see here an HLA class 1 molecule presenting what's called a neopeptide, which is derived from a mutated protein, which is derived from a somatic mutation found in the cancer cell. As this is non-self, there's a chance that the immune system will react towards it. And we see this more clearly here, the interaction between a T cell and a cancer cell and the presentation of a neoantigen. So we know that there's been an amazing success of immune checkpoint inhibitors. Some of the results are seen here, many, many more are seen, where the immune checkpoint inhibitor will release exhausted T cell to target tumor cells. We also know that in order for an immune checkpoint to work, the T cell has to recognize a neoantigen presented on the cancer cell, these neoantigens. So if there are no neoantigens, a very low number of neoantigens, there's a lower chance that an immune checkpoint inhibitor will work. And indeed, if one looks at the correlation between tumor mutational burden, TMB, and the response to checkpoint inhibitors, you can immediately see that the best responders are, for example, melanoma, which is a highly mutated solid cancer. Of course, we also see colorectal cancer, especially the MSI colorectal cancer. We see this also here, but we also know that not all patients will benefit from immunotherapy, checkpoint inhibitors, which opens the way to additional immunotherapy modalities. And you can see here various types. So we can see the adoptive cell therapy, which has been around for quite a while, where you use T cells derived from the tumor in the patient, and you expand those in order to treat the patient. But on the right-hand side, we also see vaccination modalities, for example, mRNA vaccines. On the bottom, we can see bispecific antibodies as well as T cell receptor directed against presented neoantigens. And so we'll touch upon some of these modalities. The reason I'm bringing it up here is because we're thinking that these could synergize with immune checkpoint inhibitors in order to enhance responses of patients who are not responding currently sufficiently to immune checkpoint inhibitors. Now, when we look at neoantigens, it's not only identifying them, but their quality is also very important. And I'll divide here neoantigen qualities to subcategories. We do see examples where one single neoantigen that is targeted may be sufficient in order to see a very impressive response. And I'm putting here the paper by Steven Rosenberg at the New England Journal of Medicine, where he identified a KRAS-derived neoantigen, identified T cells that are reactive towards that KRAS, and treated with this TIL, with the tumor-infiltrating lymphocytes from this patient, and saw a response. This is an N of 1 example, but it's a very powerful example because this is one particular neoantigen that is being targeted. So how are they being recognized, these neoantigens? Here's the closer look. You see an HLA molecule, HLA class 1, presenting a neoantigen to the T cell receptor. If you see a very clear and specific interaction, there's a chance that these T cells will be activated and kill the target cell. So we have a goal to create a more comprehensive atlas of cancer-specific neopeptides, otherwise known as the immunopeptidome landscape, and also try and understand better how the T cells identify these neoantigens as foreign in order to improve this personalized cancer immunotherapy. We will also deal with intra-tumor heterogeneity and how this impacts T cell recognition of tumor cells. So this is the outline. I'll first talk about intra-tumor heterogeneity, and then I'll talk about how we map the cancer cell immunopeptidome. So like I said, tumors with an increased mutational load have an increased association with response to checkpoint inhibitors. On the other hand, we also know that low heterogeneity of tumors is associated with better response. This is a paper by the Swanton lab showing that the clonal neoantigens are a very strong biomarker to response to checkpoint inhibitors. Now, we need to consider that when TMB, tumor mutational burden, is taken into account, this is bulk TMB, right? And so these mutations could be derived from different evolutionary stages as the tumor develops. It could be early on within the trunk of the tumor, or it could be later on in the branches of the tumor. And at what stage these mutations are acquired will impact the pathogenesis of the tumor. And so when you're taking the bulk TMB, you don't know at what stage these mutations are actually being formed. So we wanted to tease apart these two different attributes regarding number of mutations and intratumor heterogeneity, and how they affect how aggressive the tumor grows and how well it responds to immune checkpoint inhibitors. So in order to tease apart these attributes, Osnat and Yochai from my lab decided to expose a melanoma mouse cell line to UVB in order to increase its heterogeneity as well as its mutational load. And when these cells were injected subcutaneously into immunocompetent mice, we could see that despite the higher mutational load in these cells, they were actually more aggressive, which was exactly the opposite of what we expected. We increased the TMB. They then took single cells from this heterogeneous cell line and regrew the single cells to new cell lines, so that now the heterogeneity was low. These new cell lines were highly homogeneous, and the mutational load was random because it depended on which cell they chose. And when they injected these cell lines, 20 different cell lines, each one of them ultimately got rejected, independent of their TMB. And this is just an example of some of these tumors. They worked out that the immune system has a role to play here, because when the same cells were injected into mice that are knocked out for CD80-86, which are co-stimulatory molecules, they reverted the phenotype. And when they depleted CD8 positive T cells, they also reverted the phenotype, suggesting that the immune system has a role to play here. At this point, we wanted to start mixing the different single cell clones in order to increase their heterogeneity. But we wanted to choose these single cell clones in a rational way, not just mix them by just take 5, 10, etc. So the way we decided to mix them was based on a phylogenetic tree that was made in collaboration with Charlie Swanton and Kevin Litfield, which is seen right here. So if you identify where the different single cell clones lie on the tree derived from the UVB exposed cell line, you get this nice tree with 6 different branches. And now we can look at two fundamental components of tumor heterogeneity. One is the number of clones, and the second is the genetic diversity between them. So now we could choose these clones and mix them in a rational way. So the first set we chose was a set of 6 single-cell clones, all sitting on the same branch, so that the diversity was low. And indeed, when we mixed them and injected them, they were immediately rejected. But if we took 6 single-cell clones, 1 from each branch, so that their genetic heterogeneity was high, you can immediately see that this mix is more aggressive, despite the fact that they harbored a similar number of mutations. Now we took 12 clones, so we increased the clone number, but the diversity was low, they were all from the same branch. And again, this mix was immediately rejected. Whereas if we took 2 clones from each branch, ultimately 12 clones, and mixed them and injected them, this mix was extremely aggressive. And so you can already see the trend where you put them all in one graph. You can see that the UVB cell line is still the most aggressive. It's probably highly heterogeneous, much more than the 12 mix that I just showed you. But this is probably the most important figure, showing that it is the genetic diversity and the number of clones that are playing the role in mediating tumor growth rejection, rather than the tumor mutational burden in this particular model. So we wanted to check if this is also true for human data. And so we applied the Shannon diversity index metric to quantify clone number and diversity in different cohorts of patients who received immune checkpoint inhibitors. And so on the top, we see a low Shannon diversity index. So it means that most mutations are concentrated in one clone, versus a high diversity seen on the bottom. And when we applied this on 4 different melanoma patient cohorts, we did see significance that the tumor clone number and genetic diversity are associated with response to immunotherapy, as we saw in our mouse model. So this paper really had a few take-home messages, as described in this slide, because it has many, many more figures than what I just showed you, because we did find the neoantigens that are being presented and did look at which T cells are infiltrating these tumors. But the main take-home message was that there is a need to quantify intra-tumor heterogeneity to improve patient selection for checkpoint blockade therapy. We are following up on this study using this mouse model, also in order to develop rational cancer vaccines based on the neoantigens that are being presented on these different single-cell clones, and how these vaccines work upon tumors of different heterogeneity. And we're also looking at highly homogeneous single-cell clones that do become aggressive, and trying to understand what is the mechanism behind their aggressive nature. And the goal of that is to try and understand why patients who have tumors that are highly homogeneous still do not respond to immune checkpoint inhibitors. And those studies are ongoing. Okay, so I'll move on to the second part of my talk, where I'll be talking about how to map the cancer cell immunopeptidome. So we think that the cancer immunopeptidome reflects the hallmarks of cancer. So we have a full repertoire of MHC-presented peptides that are categorized according to the hallmarks of cancer seen here. And this is, of course, adapted from the Hanahan and Weinberg hallmarks. And so we see here, for example, genomic instability and mutations. We see viral and bacterial infections, drug resistance, etc. And we aim to capture these various hallmarks in my lab in various different projects. So I'll touch upon some of these today. And the way we map the cancer cell immunopeptidome, rather than predicting which neoantigens are being presented, we like to actually physically elute them from the cells. So we take fresh tumors or cell lines, or PDXs, we turn them into lysates, and we then perform MHC immunoaffinity purification using either PAM HLA class 1 or HLA class 2 antibodies, or mouse MHC class 1 and class 2 antibodies. We then elute the peptides with acid, and then we run them on a mass spec. And now we perform a proteogenomic analysis, which is key, because if you just analyze the spectra against the human proteome, no neopeptides will be identified because the human proteome does not have those mutated peptides. So what we do is we perform whole exome sequencing on these patients, derive their somatic mutations, and then manually add the protein sequences of those somatic mutations into the human proteome database, and only then align the spectra towards that database. And that allowed us for the first time to identify presented neopeptides, not only neopeptides, but also tumor-associated antigens. But of course, there's lots of spectra that do not get aligned because they just do not have the right sequence to align to. And we call this the dark matter. And this is actually the highest number in those spectra which are actually binned. But we think that this is a very interesting list of peptides. And so we're working hard on trying to identify what is the dark matter. More of that later. So we talked about quality of neoantigens and the fact that a single potent neoantigen may suffice to treat a cancer using a modality such as a TCR vaccine. But we also talked about clonality, which is important. The more clonal the neoantigen, the better chance there is that that tumor will probably respond. And so we're always interested in identifying clonal neoantigens, and there's quite a bit of support for this. One of the studies is the one I just told you about in our UVB mouse model. Finally, recurrent neoantigens are ideal off-the-shelf targets. So we would like neoantigens to be relevant across different patients so that those could be targeted using different modalities that are already found on the shelf. And here again is the example from the Rosenberg group, where a follow-up study by Eric Tran identified T-cell receptors that identify the neopeptide derived from KRAS G12V on its HLA molecule. And then these TCRs were used to engineer T-cells from the pancreatic cancer patient and then used to treat that patient. And this gave very impressive results. So these are the TCRs that we're interested in identifying and putting them on the shelf for future treatment options. So these recurrent neoantigens are highly sought after. One, also because they're non-disposable. So most of them are going to be derived from driver mutations, for example, oncogenes, which are non-disposable for the tumor growth. So one would want to target these because then those tumor cells are going to be killed because they need that oncogene. Also, these are going to be clonal because they are drivers, and we would make sure that they are found in the trunk of that phylogenetic tree, and probably also kept in metastases of the same patient. And of course, once a TCR is identified towards a recurrent and presented neoantigen, that TCR is transferable across different cancer patients. So when we look for neoantigens, as well as other groups, the main problem is that most of these are passenger-derived. They're patient-specific, they're non-recurrent. And like I said, we want to identify these recurrent neoantigens, so we have these off-the-shelf targets. Some of these were identified whether by serendipity or by focusing on highly mutated regions in the genome, but there hasn't been really a systematic way to identify recurrently presented clonal neoantigens. So Avia Perry in my lab derived a data-driven approach for candidate selection. And what she did is take a large database and a patient cohort, in this case a TCGA melanoma cohort. She looked for the recurrent oncogenic mutations, and she then found out which HLA allotypes these particular patients harbor. And so now she limited her analysis to the mutation HLA combinations that appear in multiple patients. And only then she predicted which neoantigens may be presented by these HLA molecules, and only focused on the most promising targets, promising being the ones that had the highest percentage of patients with that combination, as well as the ones that harbored predictions of neoantigens that are strong binders. And so we applied this methodology for melanoma, and we immediately noticed this particular target of NRAS Q61K and NRAS Q61R, potentially presented on HLA A0101. She applied this on 3 different melanoma cohorts, the TCGA, the Hartwig, as well as Australia. And in each case, we got the same exact results. So we decided to focus on both of these targets and validate wet labs, see if they are actually presented. And the way we go about that is, first of all, use a very robust system of a monoallelic B cell library that we derived in the lab. So these are B cells that are HLA null, and we overexpressed an HLA allele of interest. So in this case, it would be the HLA A0101, as well as the minigene of interest. So the actual mutated gene, but as a minigene with a mutation right in the middle. Now, these B cells are very easy to grow at large numbers, which is what we need for HLA peptidomics. And then we run them in HLA peptidomics and run the analysis in a discovery mode to see if any neopeptides are presented. And if we do identify a presented neopeptide, then we rerun the immunopeptidomics in a targeted mode, but using a patient-derived cell lines that harbor the mutation and the HLA endogenously. And we also spike in a heavy peptide, an isotopically labeled peptide, in order to validate presentation and quantify it. And so this is the methodology that we run regularly in the lab. And when we did this for the NRAS Q61K, we immediately found this particular neoantigen very nicely fragmented. And this is the isotopically labeled peptide that's pulsed into the immunopeptidomics. And we see a very nice spectra that really shows very robust presentation. This is a summary or table of the various samples that we looked at. So we have here melanoma tumors that harbor the mutation on HLA endogenously, showing the presentation. And on the right-hand side, we could calculate how much of the peptide is presented. Because we knew how much of the heavy peptide was pulsed, we could calculate how much is being presented. We could go pan-cancer because NRAS is already an oncogene that's relevant for other cancer types. So we found it presented on cutaneous T-cell lymphoma. And we also found the Q61R, which was also predicted to be presented. At this point, we wanted to see if the peptide is immunogenic. We were lucky to have autologous TIL from these patients, 2 different patients. So we could incubate the TIL at different effector to T cell ratios with the melanoma cell lines that presented the neoantigen. And we could see here percent killing that increases. So clearly the TIL is recognizing something on these cells, which is causing them to kill. But this doesn't prove that it's the NRAS Q61K neopeptide. To further prove this, what we did is use B cells that harbor the HLA A0101, and we pulsed either the wild type or mutant peptide, and again co-incubated with these autologous TIL and checked for interferon gamma release. And here you see that indeed there's an increase in the release of interferon gamma when you put the peptide on these B cells. And 17-TIL shows reactivity towards both of these neopeptides, the K and the R, which is interesting. It suggests that the 17-TIL has a TCR that's cross-reactive. So this means it's worth isolating relevant T cells and performing TCR sequencing in order to find this specific TCR. So for this, we collaborated with Neil Friedman and Ansu Sapathy and performed bulk TCR sequencing as well as single cell TCR sequencing on these TILs. And we could immediately see, even in the bulk TCR-seq, that we have alpha and betas that probably join in that form a TCR because their frequency is very similar. So even statistically, you can calculate which alpha goes with each beta. And of course, the single cell TCR-seq recapitulated this data. So ultimately, we focused on a few TCR clones that were found in these two patients, and we annotated them with a patient number, so 17.1, 17.2, etc. Some of these had 2 alpha chains, as you can see here, and 1 beta chain. So we needed to find out which alpha chain is actually forming a TCR with a beta chain. So at this point, we ordered them as RNA molecules and electroporated each chain individually to check functionally which ones are forming an active T cell receptor. So these different TCRs were then analyzed, and we have lots of figures in the paper, but I'm showing you a few here, where we, for example, took B cells, pulsed either wild type or mutated peptide onto these B cells at different peptide concentrations, co-incubated with the electroporated T cells, and checked for an increase in 4-1BB marker on the T cells, which suggests that they are activated. And you can see that the black line is the wild type peptide. It's flat, so the TCR is not reactive towards it, but the mutated peptide in red does induce increase in 4-1BB on these T cells. And here we see a TCR that is cross-reactive to both of these neopeptides, the Q61R and the Q61K. So what we really derived here is a recurrent neoantigen that can be targeted with various modalities. In this case, it's a TCR modality, but of course it can be targeted with mRNA vaccines, bispecific antibodies. And this is relevant for thousands of patients every year who harbor either the Q61K or the R neoantigen. And so we are preparing for a clinical trial to treat patients with this particular T cell receptor. But beyond that, this is an extremely powerful pipeline that we can apply pan-cancer. And we have done that, and we've identified several more of these interesting neoantigens and identified their T cell receptors. One of the recent ones is actually KRAS G12V that Shira is working on. And here you can see that not only is it presented, but it's also immunogenic, because when we use a peripheral blood and co-incubate with this wild type or mutated peptide, you can immediately see there's an increase of interferon gamma released by these T cells. And here we see it as well. And so there's 50% induction of interferon gamma, which is even higher than our positive control, which is a virally derived peptide. So the KRAS G12V neopeptide is extremely immunogenic. We performed 10x TCR sequencing and identified a single cell T cell that expanded. And so now we have this T cell receptor, which we use to engineer T cells and then validate to check if it's specific. And you can see here an example where we use this TCR to engineer T cells co-incubated with B cells pulsed with the wild type mutant KRAS. Clearly the TCR elicits interferon gamma release, TNF alpha release, as well as an increase in 4-1BB. And it is peptide concentration dependent. The black line is the wild type peptide, and the red line is the mutant peptide. So again, we have a TCR that is highly relevant for the clinic for many patients every year. In this case, also for lung cancer patients and pancreatic cancer patients, as well as others. So we're running this pipeline on various different neopeptides to find more and more of TCRs that would be relevant for off-the-shelf treatments. But we need to be aware that beyond these tumor mutational derived neoantigens, there's lots of additional antigen sources that are stimulating the immune response. And we derived this figure with Kevin Litfield, where he really integrated thousands of patient data who responded and did not respond to immune checkpoint inhibitors. And we can see that more than 50% of these responders do not have a high TMB, and they do not have an oncogenic viral infection, suggesting that there are other antigen sources that are inducing this response to immune checkpoint inhibitors. So I come back to the dark matter and asking, what are we missing? What is this dark matter, and can we identify this dark matter using other tools? So one of the dark matter components is actually derived from viral and bacterial infections. And without going to all the details, we actually did look for bacterial-derived presented peptides in collaboration with Ravit Strausman and Jennifer Wargo. And so what Adi and Shelley did in my lab is analyze 17 metastatic melanomas to which we got the 16S ribosomal DNA sequencing from Ravit for the microbial populations that are found within these tumors. And so once we had those lists of microbes that were found in these tumors, we could manually add their sequences into the human proteome database and reanalyze our immunopeptidomic data to see if any bacterial peptides are indeed presented on these different tumors. And to our surprise, we found over 200 different bacterial-derived peptides presented on these HLA molecules. These HLA class 1, and there were also over 30 HLA class 2 presented peptides. We were lucky to have autologous TILs, again, tumor-infiltrating lymphocytes to these patients from Jennifer Wargo, and we could then check whether any of these bacterial peptides elicit an immune response, and indeed a large number of them did. And so this was very exciting because it meant that there could be a correlation between the microbial landscape or signature that's found in the gut that correlates with response to immune checkpoint inhibitors and the ones that are then infiltrating tumors, entering tumor cells and being presented on HLA molecules, to which then the T cells are reactive to in the presence of immune checkpoint inhibitors. And so we are following up on this study in order to see if this could be useful therapeutically. Coming back to the antigenic hallmarks of cancer, we have a hallmark called altered response to stress signals. So this is obviously a very large hallmark, depending on the various stresses that the tumor could be exposed to. The one that we were interested in evaluating with Reuven Agami was actually the exposure of cancer cells to interferon gamma that is derived from infiltrating T cells. We wanted to see if this alters the mRNA translation under this particular stress response. When interferon gamma is secreted by the T cells, it binds its receptor and activates an enzyme called IDO1. IDO1 depletes tryptophan within the cell, which is catabolized to kynurenine, which is secreted and inhibits T cell function. But what else happens within the cell, that was unknown at the time. IDO1 inhibitors were developed, thinking that this is a new checkpoint, but these did not work in the clinic. So what Osnat in my lab did is induce IDO1 by exposing various melanoma cells to interferon gamma, and then analyze them by looking at the translatome and the transcriptome. We then analyzed the translation in these cells using DIRICOR together with Reuven Agami, who developed this tool, and immediately told us that he can see that the translation is being affected by the depletion of tryptophan. What he could also see was that not only did ribosomes stall on tryptophan amino acids, but that they then continued translating, and then about 15 amino acids later, they stalled again. And we called those W bumps. And this is really a summary slide showing a normal translation where you see ribosomes translating with equal distribution across the mRNA. But in our case, when we exposed cells to interferon gamma or depleted them from tryptophan, the ribosomes stalled on tryptophan codons, continued translating, and then stopped again. And so we, from this, gathered that the way they continue translating is by frame shifting, and then aberrant peptides may be formed as the frame shift occurs -1 and +1. You can see this in the scheme on the right-hand side, where the ribosome is shifting either one amino acid to the left or one amino acid to the right. We wanted to see if our hypothesis is true, and one way to do it was to analyze the immunopeptidome of these cells that were exposed to high interferon gamma or depleted from tryptophan, and then analyze the data for this particular hypothesis. And indeed, we identified numerous aberrant peptides presented on HLA molecules, some of which were chimeras, as you see here in black and red. What's nice is that we saw an overlap between the types of peptides that were presented in both of these stresses, and we could also see similar peptides found on metastases in these same patients. So it means that this stress response is occurring also in vivo in these metastases. And so this is a very physiological response. We wanted to see whether any of these are immunogenic, and so with Johanna Olbus and Maria Laus, they were able to identify a response to aberrant peptides using these multimers, and then when they isolated these T cells and exposed them to aberrant peptides, again, they could see an increase, in this case, CD137, which is the same as 4-1BB on these T cells. So indeed, there are some aberrant peptides that will elicit an immune response from the T cells. This is a summary of the whole process that we identified in this paper, where T cells will reach the tumor, secrete interferon gamma, which will activate IDO1, deplete tryptophan, and catabolize teichnurin, but this will also form uncharged tRNAs, which will lead to reduced translational initiation, ribosome pausing on tryptophan residues, but then these ribosomes will slip and frame shift in order to continue translations, ultimately forming aberrant peptides, some of which are presented on HLA molecules and some of which will elicit an immune response. So this is a very interesting dark matter that we identified, but mostly it opened the way as to whether we can induce the expression of these aberrant peptides in a cancer-specific manner, which of course would be very useful, especially for low TMB tumors that have a limited number of neoantigens that are being presented. And so this is an aspect that we are following up on the lab at the moment. So I showed you various different categories of presented neoantigens. And of course, there are many, many more to identify, and we're working very hard on this. And so this is just a way to show the dark matter that is found within these various different categories of presented peptides, some of which I discussed today. But again, this dark matter is extremely important for low TMB tumors. And so in my lab, we're interested in applying this, of course, in the clinic and deriving cancer vaccines. This is a beautiful figure that was recently published in Cell that I like to use, because these are the various pillars that are required in order to build this bridge, in order to derive better immunotherapies. And I hope I convince you that we're trying to contribute to some of these pillars and hope to keep contributing much, much more in the future. So this is the lab who actually did the work, an extremely ambitious and very hardworking group of people. A lot of this work was done during the pandemic, and so it was extra challenging, as well as our many collaborators that I'd like to thank. I mentioned them throughout my talk. And of course, if anyone is interested in this line of work, I have open positions in the lab, and also thanks a lot to all the sources of funding. That's it for today. I'll be happy to answer your questions.
Thank you, Yardena. That was really exciting, and so much innovative work. It's really, especially the microbial elements to the peptidome. That's just incredible. I think we're going to have the ability to. Fazia, do you want to share the screen?
I will unshare.
Oh, okay, great. So yes, thanks again for giving us this beautiful talk. You can go ahead and ask any questions to Dr. Samuels by going to Global Immunotalks and ask your questions, and we will have her reply to you. regarding what she talked about today. And our next speaker, Fazia, do you want to advertise the next speaker?
Yeah, it's going to be Kingston Mills next week.
Great. All right.
9
Well, thank
a.m.,
you
yep.
again so much, Yardena. It was wonderful.
Thank you.
And goodbye to everybody. Thanks.
Bye. Bye. Bye.
Bye.
So can you end, Governor?
Yeah.